Okay, let's talk for a minute about what we're doing today. As soon as I find my pen. So let's start defining some of these terms, and hopefully these can help us figure some of these things out. Keep those 10 facts that you had out. I'm going to collect those in a minute here. But let's talk about a plasmid. Does anyone have or I don't know why you might have heard of this, but has anyone ever heard of something called a plasmid? Nope. Good guess though? Yes. That's correct. So the definition I've been giving people all week, and I'm going to keep it consistent and give you the same definition. It's basically an extra or a piece of extra DNA. Say extra DNA that can be inserted into a bacterium. I did my best. Can you guys all read that? It's a piece of extra DNA that can be inserted into a bacterium. That's our operational definition for this. So if I were to draw a bacterium over here, get this thing. Is this a prokaryote or a eukaryote? It's a prokaryote. What does that mean? It has no organelle, so it has no nucleus, which means the DNA is just in here. All right. But it has, or it potentially has this extra thing. It has this plasmid. And a plasmid is going to look something like this. So it's extra DNA. Why do you think a bacterium might want a extra piece of DNA? What's that? It will make more copies of it, yes. But I'm kind of at a more fundamental level here. Let's back up a step. What does DNA do? It codes for protein, right? So why do you think a bacteria would want an extra piece of DNA?
Is it going to change your DNA?
Your sequence? It's not going to change. It's not going to change the sequence of this DNA itself. It's just going to be bonus DNA. Exactly. It could be for self-defense. It can help the bacteria accommodate its environment better. It can help the bacteria absorb different nutrients. Again, DNA makes protein, and protein does a lot of stuff. Okay, remember it's enzymes. It can help this bacteria digest different foods. The point is, this is like this is an add-on for the bacteria. This is the bacteria downloadable content. Okay, it adds something to the bacteria itself. When a bacteria absorbs a plasmid, it's through this process called transformation. Okay, transformation is insertion of DNA into an organism. Bless you. Okay, so there are going to be plasmids in a bacteria's environment. They're going to be out there, and when the bacteria absorbs a plasmid, that is called transformation. That's what we're doing today. Okay. So we have this other idea here, which is this idea of recombinant DNA. So recombinant DNA is DNA that is the product of multiple organisms. Okay. Recombinant DNA is DNA that is the product of multiple organisms. And I'm going to talk about what I mean there. So if we were to blow up this plasmid, for instance, I'm just going to enlarge it. We have the ability to take this DNA and insert genetics that we are interested in. Okay. Does anyone have a sense of what DNA we might be interested in putting into a bacterium? Perhaps not. Potentially, I mean, amino acids chained together are proteins. You're saying some of the right things. I can tell no one has a sense for this, but it's a really, really big deal, this process that we're doing today. Okay, so for instance, a gene we might be interested in, have you guys ever heard of insulin? What is insulin? Your pancreas produces insulin, and basically what it's for is the regulation of blood sugar. So if you happen to be diabetic, there's some issue with insulin, your body might not produce it correctly or might not respond correctly to it. And so if you are diabetic, you have to inject insulin. Where do you suppose we get the insulin for those diabetics to inject? Hearing whispering. So typically your bodies, if you are not diabetic, will make insulin. But again, if you are diabetic, there's some issue there. So if you have to inject it, where does it come from? Potentially, you could get it from a healthy body. So I guess we could farm people and like extract their pancreases, but that won't work, right? You can't just steal a healthy person's pancreas. They used to extract insulin from pork, from pigs. They would take a pig pancreas and grind it up and then extract it because they know the properties of the insulin. So that's how it used to be done. They used to take it from pork. But now what they do is they take this gene for insulin, and they insert it into a plasmid. Then they take that plasmid and they transform it into a bacteria, and the bacteria actually grow insulin for us. Pigs worked out fine for a while, but pig insulin isn't the same as human insulin, right? It's not going to be as effective. I think they still do use pig insulin sometimes, but this is just much cleaner because no one cares about bacteria, right? There are people who actually, care very much about the lives of pigs, whereas bacteria are, right? So this process is where we get insulin from. You can make a huge number of products using this kind of genetic technology, using this kind of recombinant DNA. So like a friend of mine was making spider silk proteins at the University of Pacific using bacteria. Right? So, let's see, do I have anything else to talk about right this minute? We talked about plasmids, we talked about recombinant DNA, we talked about transformation. So again, plasmids are these circular pieces of DNA that are separate from the bacterial genome. It's a separate thing. They can, in nature, pick them up from the environment. They can do that. All right? We didn't invent plasmids. Plasmids were already around, and biologists and biochemists just basically stole them. And now we use them for our own. Through the process of creating recombinant DNA, we can add our own genes to these plasmids. And then when you put them back into these bacteria, that's called transformation. Okay? So that's the outline of these are the concepts you need to know kind of moving forward with today's lab. Are there questions? I understand that this is like all of biotechnology in like 10 minutes. So there's a huge amount of detail going on here. Like, for instance, how do they get this gene in here? We'll talk about that. Or like, what are you talking about? There's just DNA in the environment? Yes. Yes, there is. So, like I said, what we are going to be doing is a transformation. We are basically going to be taking bacteria and more or less forcing them to accept the plasmid. All right, so if everybody could grab their lab handouts. We are on page 2. And this is just, again, describing the process that I was just talking about. So you grab a human cell that has your gene of interest. And by the way, every single cell in your body has all of the DNA. Every single cell in your body has every single piece of DNA that every other cell has. Which, when I learned that, it kind of like blew my mind a little bit, kind of wild. Every single cell has the complete genetic set of information. But anyway, you go in and you find the piece of DNA that you're interested in making. You find the protein that you want to make, which might be insulin, which might be spider silk, which might be whatever it is that you want to make. Okay? And you cut it out using these nifty little things called restriction enzymes. You cut it out using these things called restriction enzymes. And restriction enzymes they're basically just molecular cutters. What do they cut? They cut DNA. Restriction enzymes are these molecular cutters. They cut DNA. So you grab a cell, probably grab a lot of cells. You cut out the gene you're interested in using those restriction enzymes, right? And you cut the plasmid open with the same restriction enzymes. And basically what you have is something that looks kind of like this. You got your plasmid here. You've got your insulin gene here. And you take this and you paste it in. You cut and paste this gene into the plasmid. This part of the process is called ligation. This has already been done for you guys. I have plasmid that's just kind of ready. So you insert this plasmid back into your bacteria. What's that step called? Transformation. Okay. What's that? That's right. You're taking your DNA, you're inserting it into your bacteria. Transformation. And then you let the bacterial cells grow. And they are going to make your gene that you're interested in. They're going to make your insulin. I didn't ligate this in yet, but it's in there, okay? So we are starting basically in step 2. We're doing the transformation. Like I said, the ligation has been done for you already. Let me check my plasmid. Got plasmid for you right here. So let's go through the protocol. All right, let's go through the protocol. That's on the next page here. Step one, and you take some notes, I would say, just alert yourself whenever you think you might need that. And there's one step in particular where I want everybody to put exclamation points and circle it and highlight it and stuff. But step one is label one closed micro test tube plus P-glow and the other minus P-glow. Label both tubes with your group's name, place them in the foam rack. Step two, open the tubes and using a sterile transfer pipette, transfer 250 microliters of transformation solution, CaCl2. Step 1 and 2 have been done for you by Devin, the guy that makes me look good. Okay. Well, for this lab anyway. Sarah is usually the one that makes me look good, but for this lab it's Devin. Step 4, use a sterile loop to pick up a single colony of bacteria from your starter plate. So let's unpack that a little bit. So here are our starter plates right here. Let's find some open loops. Devin did too good of a job. There are no open loops. All right. Here are our bacteria. They say E. coli on them because they are E. coli. All right. And it says grab a single colony. And there are millions and millions of bacteria on this plate. You basically just need to get a little bit of them. Okay. You just need a little bit of bacteria, and you're going to put it into that transformation. Okay. It even tells you how to roll the thing between your fingers. All right. And I'm going to need to get a new red bag, but when you're done with your stuff, anything that touches bacteria needs to go into this red bag. Once I find Devin and give me one. Just saying, it makes me look good, and it's making me look good. All right, so we're doing that for the plus and for the minus. Now, step 5, blah, 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 blah. We're not doing that. You're bringing it up to me, and I'm going to be using this pipetteman to add 10 microliters of plasmid to just your positive tube. Just the positive tube. So step 5, you can kind of cross that out and just bring it up, just write and bring it up there. Step 6, incubate tubes on ice for 10 minutes. And this is another modification. It says make sure the tubes are all the way down in the rack so the bottoms of the tubes stick out and touch the ice. Don't do that. Just put the tubes directly onto the ice. So you're going to have these little foam test tube racks. These do not need to go on the ice. You can just put the tubes directly into the ice. Okay. Step 7. While the tubes are sitting on ice, label your agar plates accordingly. We're going to talk about the reason why we're doing each of these steps at the very end. Okay, we're going to discuss sort of what each plate means and why we did it. Step 8. This is the step that needs some exclamation points. This step you need to be very aware of what you're doing. Okay, it's not hard. You just need to pay attention on this one. Okay, you need to put it in our hot water bath for exactly 50 seconds. Not 45 seconds, not a minute, 50. 50, okay? And when you do this, you can take your ice containers with you, because those are full of ice. You can just bring them with you over to the incubator, because the shorter the amount of time between when it gets heat shocked and when it goes on the ice, the better. Okay? The less time it spends between here and the ice, the better. So again, if you were to heat shock it and then walk it back to your desk, that would probably be fine, but we want to optimize this. We want to get the best results that we can. All right, did everybody put exclamation points or happy faces or whatever makes you pay attention there? Step 9, remove the rack containing the tubes from the ice, place on the bench top. Open a tube and using a new sterile pipette, add 250 microliters of LB nutrient broth to the tube and reclose it. Repeat with a new sterile type pipette for the other two. Incubate for 10 minutes. All right, so LB will be in the tube that's labeled LB. Demonstration. And it says 250 microliters. All right. What that means is we have these transfer pipettes here. They're hermetically sealed, and so you kind of need to find a little flap. It's like a string cheese. Open it like this, and it says on the very top 1 ml, right there, 1 milliliter. 1 milliliter is 1,000 microliters. So if I wanted to get 500 microliters, I go to the 5. If I wanted to go 250 microliters, I would go down a line. Does that make sense? It feels sketchy.
So the first line.
This is 1,000 microliters.
First line from the bottom.
Mm-hmm. Yep. This is 500 microliters. This is 250 microliters. There's a little picture on your lab even that kind of shows points to the appropriate line. But it is tough. Some folks have never done this stuff before, so I want to make sure. You know, if you get confused about it, just ask me, no shame. I'll be pretty discreet. All right. So again, 1,000 microliters, 500 microliters, 250 microliters here. And you'll be doing the same with 100 later on, and it shows you in the lab exactly what to do with 100. Okay. After you transfer 250 microliters of LB nutrient broth onto or into those tubes, you're just going to let them incubate. And then you're going to transfer 100 microliters of the transformation and control suspension onto the appropriate place. So again, protocols always sound worse if you just read them out of context. This should make a ton of sense once we actually get there, if you're lost or confused or whatever. And then with a new sterile loop, spread the suspensions evenly around the plate. So what you're going to do is you're going to take a new sterile loop, and you're just going to spread the bacteria that you put onto the plate around. That's it. You don't need to stab into the agar. The agar should not be deformed by this. You just want to spread out the bacteria that you've added to the plate. All right? Then you stack them up, tape them together, and you give them to me. So a word about these E. coli. These are what are known as K12 E. coli. These are non-pathogenic. All right? They will not hurt you. Please treat them with some respect, though. Do not lick them or anything like that. Not just saying that for your benefit. Just, again, wash your hands.
Where'd you get this coli from?
Biorad, same place we got the plasmid. It's basically like a store. Anybody could buy this stuff. Just be like, I'm a teacher, and they'll sell you pretty much anything. Yeah, it's wild. Wow, you flip through the catalogs and you're like, they have that? Yeah, they've got that. So that's like the whole protocol. Make sure you give that a read before we get started. Are there questions before we get going? I know that was a lot. Yeah, a lot, especially for a Friday morning, right? I'm sorry. I didn't pick this lab list. All right, so again, you can do it by yourself or you can work with a partner. I would recommend kind of most folks doing it by themselves. It's up to you though. We've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 14. It's like 16 or 18. We have a bunch of setups. So if a couple people work in partners, everybody else can do it individually. But it is up to you. It is up to you. So go ahead and get going. I will be here for questions and stuff. You can do this experiment all right along.